I am devoted to be the most boring part in this lecture because I will give you the basic principles and the basic application of the atomistic molecular dynamic simulation, while my colleague Domenico Marson will give you a wider panorama in which he will give you more advanced application of molecular dynamic simulation. This is the outline of my presentation. I will start with a general overview on atomistic molecular simulation and its definition. Now I will focus our attention on the basic concept of force field, the main and fundamental part of atomistic simulation, and the main branches of atomistic molecular simulation, that is molecular mechanics and molecular dynamics. Finally, I will give you briefly three different application studies providing from my research, in which I will give you the potentiality of the application of molecular dynamic simulation in biological system, with example about the interaction study of respectively drug-protein complex, protein-protein complex, and nanovector complex. Let me start to see that atomistic molecular simulation is just a part of the entire multi-scale molecular simulation techniques. In fact, molecular simulation is a multi-scale techniques. To give you this example, you can think about an analogy with the optical techniques, in which with the lens you can see you can see a sample with not so high level of detail, but you are able to see a big amount of this sample. While on the completely other field, you can use electron spectroscopy for CEM or TM, in which you can reach an extremely high level of detail, but you are confined in a very, very tiny sample about the system you want to study. You can use this analogy also for molecular simulation, in which you can have higher scales, in which with which you can simulate larger system for a lot of time in microfem and coarse grain. And if you wanted to use, if you wanted to get inside a more specific detail, more high level of detail, you have to use a lower scale, for example, atomistic and QM, the quantum mechanics scale, in which you are able to describe the molecular system atom per atoms, or even better in QM, also consider the subparticle, electrons and so on. But you have to pay a compromise in which you can perform this kind of simulation with decisively smaller molecular system and for phenomena lasting less time. In any case, I will focus my presentation on the atomistic simulation, because this kind of scale at the molecular simulation offer the best balance, the best equilibrium of molecular details and dimension of the system for a biosystem. Molecular simulation, atomistic molecular simulation is a software representation of a real molecular system. So we have to start from a real system, providing from experimental or general biological question. So we have to answer some experimental question. To answer this real experimental question, we have first to create a representative model, a starting structure about our molecular system. So we have to define the architecture of our molecular system, and we have also to combine this representation also with a mathematical formulation. So we have to find quantitative descriptors able to be managed by a software, a computational system. After this combination of representative model, mathematical formulation, we have to apply well-defined atomistic molecular protocols in order to predict important properties and retrieve important information about the molecular system that we want to study in order to answer the experimental questions. Why we need the mathematical information? Because we have to translate the chemical information into numbers in order to be calculated and simulated. For example, just to give you an example, if you see this molecule, this is a completely random molecule created by from scratch. Even with the very basic knowledge about organic chemistry, you are able to say a lot of things about this structure. For example, you are absolutely in grade to describe how many carbon atoms have this structure, how many oxygen atoms, hydrogen atoms, or nitrogen atoms in this structure. You are also able to discern between a single bond and an aromatic or a double bond and something like that. And you are able to understand that these, for example, carbon atoms, aromatic carbon atoms, is completely different from these carbon atoms involved in a double bond with oxygen for chemical and physical point of view. So even with a very low knowledge about organic chemistry, with a very basic knowledge, we can define a lot of qualitative properties of this molecule connected from what? Connecting from their structure, so the geometric description, and connecting with the functional groups. We have to translate, if you want to apply computational atomistic simulation, we have to translate this qualitative information into numerical description in order to take in account geometric description, connectivity, bond, angles, torsion, molecular conformation, and electronic description, atom types, partial charge, electronic force, and something like that. We have to find a numerical way in which we are able to describe quantitatively this kind of structure. In quantum mechanics, we solve this problem by apply the Schrödinger equation, or better, the approximation, the Born-Oppenheimer approximation of the Schrödinger equation. And with the Born-Oppenheimer approximation, we are able to describe the energy of the molecule connected to the coordinates of their nuclei, to the nucleus of the atom, to the position of the atom, and the geometric aspect of the atoms. This is the most accurate, the most precise way to simulate a molecular structure, but it's also the most expensive in a computational resource point of view. So we are able, obviously, to describe, to apply quantum mechanics, but with a smaller system. If you want to increase the dimension of the system and treat with molecular simulation the bigger system, such as the typical system of nanobiological system, for example, protein system, acid nucleic system, membrane system, or something like that, we have to apply another approximation and switch from quantum mechanics to appropriately to the proper atomistic molecular simulation. And we have to perform here important conceptual approximation in which we have to define the atoms as particles, that is, as rigid spheres. We consider the bonds connected to each atom as springs, and we have to treat the non-bonding interaction, the interaction between the non-bonded atom, with simple potentials. With this approximation, we are able to define the energy of the molecular system connected with its structure by a sum of different equations of defining the energy, providing from the classical physics. And this is exactly the concept of the force field. Force field is a compact model, is the base of the atomistic molecular simulation. Force field is the compact model able to calculate the potential energy of molecular system as function of its structure. So first of all, we have to, now we find a numerical, the force field is the numerical approach defined by the atomistic molecular simulation. In a force field, we have the definition of atom types. That is the discrimination not only between among different elements. A carbon is different from an oxygen or a nitrogen or a nitrogen, but also the different atom types. That is, with the force field, we are able to discriminate between different kind of carbon. sp2 carbon is different from sp3 carbon. An aromatic carbon is different between respect another carbon atom or something like that. Then a force field must contain an equation able to consider the energies underlying the geometrical and electronic aspect. And now I will give you more detail about that. Obviously, a force field must contain also the parameters that allow us to solve the question underlying the different energies. To get in more detail about that, the force field energy is composed by two main blocks. The geometrical block, in which we have bonded energies, the energies involving atoms directly chemically connected each other, and we have the bond stretching, two atoms connected each other, the angle bending, three atoms, the angle between two different bonds, and the torsion movements. These energies relied in the bonded energies. While we have the other main block, the so-called non-bonded energies, the energy in which two not chemically connected atoms are involved, and basically they are described by the van der Waals and electrostatic energies. We have to find an equation able to describe these energies, and we have to find appropriate parameters able to solve this equation and describe it quantitative the structure of a molecular system. To do this, we get in more detail. For example, about the geometric aspect, I put here the three different energy: bond stretching, angle bending, and torsion. And look at that. For example, the basic approximation is that atoms are spheres, are rigid spheres, connected each other by springs. So the energy of the bond stretching could be simply described by the spring law, the Hooke law, in which we have a product between a constant and a different in length, in which the different regard the actual distance between two connected atoms, and R0, the equilibrium distance of representing the respective atom. What does it mean? It means that if I want to solve this equation in the force field, I have to find the exact value of Kb, the constant of spring, and the R0. And this Kb and R0 obviously depends of what? Of the kind depending on the kind of bond. A single bond will have different Kb value respect double bond, aromatic bond, or triple bond, but also it depends of which kind of different atom types are involved in the bond. So we will have a different Kb or R0, the equilibrium distance, on the basis of which kind of different atom types will be involved in this specific bond. R obviously is the variable that could vary during the molecular dynamic simulation. With the same concept, you can apply the same concept also to understand the angle bending and the torsion movement. Torsion is particularly important in larger system, in biological system, because they underline the conformational changing of the molecule. But in any case, we have to define a question, the question describing the energies, and we have to find the parameters inside the force field to find to solve dynamically these equations. Obviously, this is I take the occasion. It's worth to mention here that we don't have just one simple force field valid for all the chemical environment, but we have a lot of different force field specific for a specific chemical environment. For example, we can have a force field very specific and very accurate for protein, and another force field very specific and very accurate for, let's say, a polymer system. Why? Because constant and equilibrium distance could be different depending on the molecular system in which we have to study. So we don't have just one force field in atomistic simulation, but we have to choose appropriately the best force field, and you will see better in the presentation of my colleague Domenico Marson. It's extremely important, probably is the most important choice that a molecular simulator has to have to do before starting the simulation. The choice of the best force field is the most important choice for atomistic the molecular simulation. Further, in order to conclude this point, the force field, the energy of the force field, I want to conclude with the non-bond term. In non-bond term, we have the energies involve atoms physically interacting each other, not chemically connected, but physically interacting. And we have two kind of, generally we have in the force field two big ensembles defining this kind of interaction. The van der Waals force field, the balance between repulsion and dispersive forces, and the electrostatic force field. Even in this case, we have an equation. For example, in the case of the electrostatic, the equation is trivially the Coulomb law, and we have to find in the force field, or with other ways. In any case, we have to find the parameters that we will allow to solve this equation during the molecular dynamics simulation. For example, since the electrostatic contribution is given by the product between the two partial charges of atom 1 and atom 2, we have to know the partial charges of the atoms involved in this kind of interaction in order to define this polar interaction. Then, so we find with the force field a mathematical formulation to describe our molecular system. So we have a lot of equations describing the energies of my molecule based on the coordinates of the nuclei of the atoms. Now we have to treat this potential, and we treat in two different ways. With molecular mechanics, it's a specific branch of the atomistic molecular simulation, in which we can modify statically the coordinates of the atoms in order to find a geometrical optimization. And then, more important for this presentation, molecular dynamics. That is the opportunity to insert the time parameters and temperature and the other physical parameters inside our simulation and create a real molecular system with our atomistic simulation. First, before starting a real atomistic molecular simulation, we have also to define, first of all, we have to define which kind of model we want to use to simulate our molecular system and to retrieve appropriately their properties. We can work in isolated model, in which we focus the attention on the molecular complex itself, but especially in nanobiological system, what we want to study is the molecular behavior in surrounding by the physiological environment, for example. And so we have to create a bulk model, including solvent, ions, and other boundary conditions. So if you want to simulate physiological like condition in atomistic molecular simulation, we don't have to hold only in the isolated system, but we have also to build the surrounding environment of the molecular system. For example, a protein acts in a water surrounding, or a membrane protein work inside the membrane. So we also add these components in our molecular dynamic simulation in order to reach or to be closer to the physiological-like solution. Defining this preliminary situation, what we need to run a real simulation. We need all the things that you find in this slide. So we have to start with the coordinates, the initial coordinates of our molecular system. We have to find a way to build the three-dimensional structure of our initial molecular system. Now, with the force field, with the choice of the force field, we have to describe the topology of these coordinates, and we have to create the environment, the physiological environment of our system. After this preliminary step, we have to define appropriately a computational protocol that allow me to build a atomistic molecular simulation, and from this atomistic molecular simulation, we will able to retrieve, through specific analysis, to retrieve the important information that we have to connect for describing an appropriate properties to solve the question provided by the experimental by the real system. Let's start. What we need a starting structure, which is, and this is probably the first critical choice. Why? Especially in the simulation of a biological system. Because if we want to simulate just a small organic molecule, this is not a problem, because you can draw from scratch the molecule, and then you can apply the protocol we see previously in the presentation. But in the case of biological system, if you want to simulate a complex in which are present a protein, a membrane, or a nucleic acid, or something like that, this is not the choice. This is not an option. We have to find a starting structure in different way. We cannot build by scratch the structure of a very complex situation like in the protein system. And we have to exploit some options, for example, by exploiting experimental techniques able to retrieve us the three-dimensional coordinates of our molecular system. For proteins, for example, it's very common start from the crystallographic structure, or start from structure provided by electron microscopy or NMR structure. Basically, the most common situation in the actual situation is starting from X-ray crystallography. There you can find a lot of crystallography data appropriately in different databases on internet, and you can download the three-dimensional coordinates for this of your structure. And this will be the starting point of your atomistic molecular simulation. In the last years, with the breakthrough of machine learning and artificial intelligence methods, we had also other robust option provided by computational techniques, for example, homology model and AlphaFold server that are able, from the basic amino acid sequence, to provide by computational methods the initial structure of your protein. Keep in mind that the initial structure of your protein is just a starting point. We have to apply atomistic molecular simulation in order, first of all, geometric optimize this structure, and then to move by the molecular dynamics the structure in order to dynamically simulate the chemical behavior in the real environment. Just a more specific point, if we want to study, for example, the interaction between a biological system such as a protein and a small molecule interacting with it, in the case, for example, of drug-protein interaction, it's not so common that we are able to find an experimental complex to use the three-dimensional coordinates. What we can find is the three-dimensional coordinates of the protein. We have to create by scratch the three-dimensional proteins, the coordinates of the drug, and then we have to apply very specific techniques named docking in order to create a well-weight with a robust initial structure in which the drug is inserted not randomly inside a protein, but the drug will be inserted specific in the most probable binding site based on the energy behavior by exploiting this kind of techniques. So the docking techniques must be applied when we have to find a robust position, not by random or by manual constriction, but a robust position of two different molecules. And normally, the most common option is the so-called protein-drug docking or protein-protein docking. And this is the initial, the techniques, the docking techniques, is a technique that will allow how to find the initial structure of a complex and not only of an isolate molecule. So, atomistic molecular simulation start always with the molecular mechanics. Why? Because first of all, we have to perform energy minimization. Independently from which kind of source you obtain the starting coordinates, these starting coordinates are not optimized. So we have to apply a mathematical approach in which we have to apply a minimization algorithms able to modify statically the coordinates of the system by applying the force field equation and find a decrease in the energy of the system. So the starting structure of your molecular system is not the real structure that you will move with molecular dynamics, but it's just a preliminary structure that you have to optimize. And you can optimize by applying energy minimization through molecular mechanics. Molecular mechanics solve the energy equation of the force field, exploit the equation of the force field with the minimization algorithm to obtain a geometrical optimization of your molecular system. And this will be the real starting point of your molecular dynamic simulation. Keep in mind, this is an energy minimization, it's just a geometrical optimization. We have no time, we have no temperature in energy minimization in molecular mechanics. We have no dynamics. To study biomolecular function, we must introduce motion. We must introduce a kinetic effect in which we move atoms, and we must introduce the concept of molecular dynamics. In molecular dynamics, we add time in our molecular system. Atoms will move in response to the forces generated by the force field, and will move by external forces, for example, temperature parameter. What we do in molecular dynamics? In molecular dynamics, we are able to move deterministically the atoms of our molecular system by applying equations of motion, able to give me to build atomistic trajectories of our molecular system. What does it mean? It means that at the time zero, at the starting time, we have a definition of our system not static. We have a definition of our molecular system in which each atoms each atom is defined by 6 values. 3 values for coordinates, x, y, z coordinates, and 3 values for velocities. The velocities in x-axis, y-axis, and z-axis. What we want to do during molecular dynamics simulations is build a trajectory. That is, we want to know new positions and new velocities after a specific delta t. Delta t, that is the time frame in which we have to solve the another equation and build deterministically the atomistic trajectories. Molecular dynamics engine will compute forces and from the force field with updated position, so we'll assign new coordinates and updated velocities by assign new acceleration, and they save trajectories over the time. So we start to build a atomistic trajectory. What is the most important concept, a parameter that we have to define to build a robust molecular dynamics simulation? First of all, we have to define the time step. The time step is the delta t between in which we want after which we want to know new atoms coordinates and new atoms velocities. So this delta t must be significantly low because the time step had to take in account the physically phenomena during the molecular trajectories. Physically phenomena, for example, the bond stretching, the bond bending, and something like that, this kind of phenomena last not second, not microsecond, but last very, very tiny small time in the range of femtosecond. So if we want to make integration possible and we want to take in account the physical meaningful of our molecular dynamics simulation, we have to define the time step in the range of the femtosecond. Just to recall, femtosecond is 10 at the minus 15 second. And what does it mean? That in this range, normally in the range of 1 or max 10 femtosecond, most often in the range of 1 or 2 femtoseconds, we want to know new positions and new coordinates. Try to think about this value, how many operations the software have to perform in order to create a robust simulation, a robust atomistic molecular dynamics simulation. Just to understand the complexity of the system, let's say, for example, we want to describe a biological phenomena. A phenomenon in which we have to describe the interaction between the two proteins, for example. Now it's a common system, but in order to acquire robust statistics from this kind of phenomena, the total simulation time must be in the range of microseconds, for example, in order to have a good statistic and define and describe appropriately this kind of biological system. Okay, we have to perform 1 microseconds of molecular dynamics simulation. My time step is 1 femtosecond. So if I want to perform 1 microsecond of a trajectory, I have to perform 1 billion of molecular dynamic steps. In each steps, each atoms must be in each atoms. For which atom I have to assign 6 different values to define our molecular system. So just to understand how many operations are needed to create an atomistic molecular structure. This is just a benchmark in order also to understand practically what is the time to acquire a good molecular dynamics simulation. This is a system of more or less 100,000 atom, so a system composed a medium large biomolecular system. And with a software named GROMAX and with a computational resource of one GPU, the best performance is reached in this with this kind of GPU, in which we have to wait 4 days to acquire 1 microsecond of simulation. It could be a very long time. It seems a very good time in order just to have 1 microsecond of simulation. But if we come back 10 years ago, this time was impossible to think because we don't have this kind. We did not have this kind of computational resource to perform this kind of calculation. Now, this 1 microsecond is, let's say, habitual. It's quite common to perform an atomistic simulation. 10 years ago was one thing that seems impossible to reach with the previous molecular resource. Keep in time, okay, another important parameter that we have to set, taking account during molecular dynamics is temperature, because temperature is directly connected to the kinetic aspect. Temperature will drive the motion of the atoms, so will give exactly the kinetic term through the Maxwell-Boltzmann distribution. And in order to keep constant at the desired temperature during the molecular dynamics simulation, the set temperature, we have to apply the so-called virtual thermostat. So the molecular simulator have to choose appropriately the best thermostat to keep constant with no so important variation, the temperature during all the molecular simulation. Why? Because if the temperature will have so many discrepancies during the molecular dynamics simulation, we are not able to calculate to define an equilibrium in energetic values, because this kind of variation will affect directly also the energetic term. So the temperature must be well controlled during the molecular dynamics simulation in order to make motion physically meaningful. Other two important constant parameters that we have to set for defining your molecular system is the definition of the periodic boundary condition. That is what you create at the beginning of the structure of the simulation, and also before starting simulation, is a confined space in which you put together your molecular system, for example, a protein and a drug, and the surrounding environment, water molecule, ions, and something like that. But you create a confined space with confined dimension. During the simulation, if you not apply appropriately condition, some molecule can escape from this box and disturbing and disrupt completely the molecular dynamic simulation. What you can do to avoid this opportunity, you have to apply this condition. You have to apply a bulk condition in which you avoid artificial walls, you reduce the surface effects, and you are able to mimic a bulk solution. The periodic boundary condition is a computational techniques able to reproduce virtually the original cell, the cell you created, the box you created at the beginning, in all directions, in order to create a virtually infinite bulk system, without affecting directly the computational resources. Why? Because the other cell, the virtual box, are exactly as the original box you created. And what happened in the original box will happen also exactly in the same way in the reproduced boxes with the periodic boundary conditions. So if you work with let's say if with an environment surrounding your molecular system, if you want to work with a bulk model, you have to apply periodic boundary conditions. And finally, if you want now then to retrieve some important information about your molecular dynamics, you have to define a statistical ensemble that define the thermodynamical description that have the thermodynamical description of your molecular system. You have to fix some physical parameters. The physical parameters you have to fix normally are the number of atoms, the volume of the system, the energy of the system, the temperature and the pressure of the system applied during the molecular dynamics, and you can apply different statistical ensemble in order to reach different molecular dynamic situation and obtain different level of information and different way in order to extract appropriately some properties. Here I put the most common statistical ensemble that you can define, NVE, NVT, and NPT. This is a very specific, it's quite more specific respect the other information. So if you want to have more clarification, I will be happy to give you more clarification about that after my presentation. In any case, the common workflow of molecular dynamics is first minimize the starting structure with molecular mechanics, then perform an equilibration by exploiting an appropriate statistical assembling, and then run the production and build the real atomistic trajectories by specify appropriate statistical ensemble to retrieve information from this statistical ensemble. This is quite a very basic schematic approach in which we can obtain some important parameters from atomistic simulation. This is a common scheme that we can apply for the study, for example, a drug protein system. First of all, we have to find the initial coordinates by docking study. We optimize the result of the docking by applying molecular mechanics. Then we have to apply molecular dynamic simulation in order to understand what will happen between protein and drug during time and during the molecular dynamics trajectories. And then at the end of the simulation, we have to retrieve the coordinates and apply some analysis in order to obtain information about context, structural information, and energetic information. For example, this is a normal example. The raw data from the molecular dynamics is quite unusual from without applying any other analysis. If you want to obtain a robust analysis characterized with the properties of your molecular system, you have to apply other kind of analysis, because from the raw data of molecular dynamics, what you know is just the position of your atoms and the velocity of the atoms. You have to apply specific analysis that you will able to retrieve important structural and interaction information to define your molecular system. In this slide, I report here the most useful observable parameter that you can obtain from molecular classic and common molecular dynamics simulation, and to answer directly an experimental or biological question. By applying specific analysis on the molecular dynamics trajectories, what you can obtain is, for example, the description of the distance and contacts between atom or group of atoms. So you can hypothesize some contacts or some interaction between atoms. You can get more in detail about this kind of information. For example, you will be able to define H bonds or salt bridge between two specific atoms among different groups. You can also apply structural analysis in order to clustering the structure and obtain the most common information about, for example, the biological and so on, the biological active conformation of biological system, for example, between protein-protein or between protein and drug, but by applying specific energetic analysis. In this slide, I report one of the most common, the so-called MMGBSA analysis. You were able to quantify the binding energy trend. That is the binding energy between molecules inside your molecular system. And now I will give you some example provided by my research in order to understand better the potentiality of this approach. Let's say what I'm able to perform with molecular dynamics in biological system. We are able to answer or to predict, to answer a experimental question or to predict molecular protein, to predict molecular properties. For example, I will report here three different cases. One drug protein, another protein, and the final nanocarrier protein, in which we have to answer different experimental questions. In the first one, we have to answer where and how does the ligand bind, which is the position of the ligand inside the protein. In the second one, which in the protein-protein system, which will be the residues that must stabilize the recognition between two proteins. In the final examples, you will see how the molecular dynamics simulation can be used in order to obtain the description, the interaction between nanocarriers and biological system inside our organisms, for example, plasma proteins. Let's start with the first one. In any case, we can connect all the three examples with one general question. The common question is which molecular interaction control the observed behavior? Obviously, the answer depends on which kind of the molecular system we want to study. In drug protein, I will lead to you the example about two different anti-cancer drugs, dabrafenib and emburafenib, in binding the most abundant protein of our organism, that is the human serum albumin protein, the HSA protein. What we have to answer, what we have to explain with molecular dynamics. From experimental techniques, calorimetric and fluorescence techniques, we were able to calculate, to measure that dabrafenib and vemurafenib bind HSA with the same affinity, but with a completely thermodynamic approach. Dabrafenib bound the HSA with an entropic-driven approach, entropic driven binding, while on the other hand, vemurafenib binds HSA with an enthalpy-driven binding. How is possible? We have to get in molecular detail this kind of question. We have to answer this kind of question with molecular detail, and we have to explore molecular dynamics. So we adopt a classical approach in which first we dock the ligand into the protein, then we minimize the molecular system, and then in the solvated environment we perform molecular dynamics, and from molecular dynamics we were able to differentiate at atomistic level the binding way of these two drugs. The dabrafenib, the binding of the dabrafenib HSA complex is dominated by hydrophobic contacts, and we are also able to define the most involved amino acid in the contacts, while vemurafenib binds HSA by performing hydrogen bonds and a lot of polar contacts. Hydrogen bonds and polar contacts are specifically from enthalpy contribution and is a more directionally it's defined as a more directionally anchoring inside the HSA protein. Even in this case, at atomistic level, we were able to highlight the most interacting residue inside the system. In the second case studies, we enlarge the system. We treated the protein-protein system. This is a work about 5 years ago during the COVID period. Unfortunately, we had a lot of time to perform simulation at period because we had to stay at home. And so we decide to describe at molecular level the interaction between the spike protein of the virus and its human target, the SA2 protein. That is the first protein in contact with the virus during the recognition of the virus inside the human body. We perform a lot of molecular dynamic simulation, almost 200 of different replicate molecular dynamic simulation, in order to first define the interface between these two proteins, which are the spike amino acids most involved in the binding, and which are the AC2 amino acids most involved in the binding. And then we perform a step further, and we mutated all the most involved amino acids of the spike protein into the other 19 amino acids in order to try to build a predictive map of the potential risk of each mutation. That is, the potential risk will mean a mutation will increase or will decrease the binding between spike and AC2. If the binding will increase, the mutation is more risk. Otherwise, if the binding will decrease, the mutation will decrease the risk of interaction. We applied also an experimental validation with external experimental group, and we obtained with our molecular dynamics model, a correspondence of about 90% between our prediction and the experimental values. And in these articles, you will find some hotspot residues that after 1 or 2 years effectively actually will have been shown in the more risky variants of the COVID virus. Sorry.
Sorry, 5 minutes till the end.
Yes,
please.
Perfectly in time. Perfectly. Thank you. Thank you so much. So this is another example in which, by application of the molecular dynamics, you will reach details that you are not absolutely able to reach with other techniques. And in this case, I had not only answered a question, but we perform also a predictive model to try to understand the risk of the plausible mutation in a specific protein environment. Finally, the more complex case study in which we have a macromolecular system composed by micelle, by a micelle of a nanovector. This is the structure of a monomer of the micelle. This is an amphiphilic nanovector used for the drug delivery and for the gene therapy to deliver nucleic acid. But what was of our interest, it's not the interaction with the target, but the interaction with human proteins, the proteins most abundant in our blood system, because these kind of proteins dramatically affect the biodelivery of this nanovector system. So what we use is in this article is the combination between experimental and computational molecular dynamics approach in order to obtain a molecular level description of the interaction of these micelle, the nanovectors of these micelle, composed by this mice, and the HSA. From the experimental techniques, we obtain the thermodynamics of the interaction and enthalpic and entropic balance. So for binding nanovectors and HSA proteins. With DLS and electronic microscopy, we obtain the dimension and the structure dimension and the shape of this system. And we exploited this information to build our molecular dynamics complex. The atomistic complex, you can see here an image in which you have the mice as represented by these light blue spheres, and they're surrounded by 6 different HSA proteins. So we are able to describe the first shell of soft corona environment at atomistic level. With molecular dynamics simulation, we were able to corroborate the data, the results obtained with experimental value, but we were also able to give a molecular image about the complex formed by nanovectors and HSA and albumin proteins. Moreover, we were able to get more in detail by applying specific energetic analysis. We were able to define the electrostatic interaction between each albumin and the nanovectors, and also to retrieve, to describe the key amino acid regions inside the albumin in order to create an amino acid map and to see which kind of residues are more connected and more involved in surrounding and absorb into our nanomices. Finally, the final message from the description of these 3 different cases is that molecular dynamics is one of the most powerful tools that you can use as an interpretative bridge between experimental and molecular mechanism and computational mechanism in order to obtain not only information but also structural description and images at the atomistic level, quite impossible to obtain with any other experimental technique. This is the end of the first part of our presentation. This is the end of the presentation. I want to thank you for your kind attention, and then I want to add a special remark by my personal, as a personal note. I wish to express my solidarity with the entire Ukrainian people during this very difficult time, with the hope that the war may end as soon as possible and that a long-lasting period of peace will follow. Thank you again, and I'm open for any question you will solve. Bye-bye.
Thank you very much. Thank you very much for impressive lectures, and especially thank you for your solidarity. It's very important for all people in Ukraine. And now the session of questions is open. Please, if you'd like to ask something, maybe somebody from Zoom audience. If people still are thinking, I will ask. For me, Erik, it's very interesting lectures because I'm a biophysicist, and in our department of molecular biophysics in the Institute of Low Temperature Physics and Engineering, we are really investigating nanosystems, interaction with biomolecules and with drugs molecules. And that is why your examples, your presentation was really interesting for me. And my question is, for example, for the last system, which is the last example of a complex of nanocarrier with human serum albumin, as I understand, which type of experimental you used to confirm the complexation? Could you pay more attention?
Okay, yes, basically it's okay. Can you hear me?
Yeah. We hear you. Thank you. Please stay.
Okay, let's say we use a proxy evidence because we have not a direct information about the real number of how many HSA molecules bind the nano vector, but we use a combination of different techniques in order to obtain a reliable and a robust number for describing this kind of complex. First of all, with calorimetry, we are not able to just to have a thermodynamic description of the binding, that is not only affinity and the mechanism, the thermodynamic mechanism, but if you are able to set appropriately the experiment, you can also able to retrieve the stoichiometry about the interaction between the molecule involved in the experiment. And so from the ITC experiment, we are able to define a stoichiometry about from 5 to 7 HSA involved in the system. Combine this information with the dimension and the shape obtained by TEM and the TEM and the DLS, the dimension of DLS, we perform a sort of initial, let's say, a starting system of molecular dynamic simulation in which we have first the nanovectors surrounded by 5 albumins, then 6, and then 7 albumin. So we simulated the 3 different situations, providing by the combination of the information providing from the calorimetric and then TEM and DLS techniques. And what we see at the end of the simulation, that is the most robust, the most probable system, was that composed by the combination of HSA, 6 HSA and 1 nanovectors. And this is just I hope that I answer your question in define the initial structure.
Yes, thank you very much. But also I propose to use maybe a spectroscopic or mass spectrometry method really to confirm that all the protein and nanocarriers is involved in the complexes formation, because microscopy or DLS is just approximation of the size of the complexes, but not confirmation the content, which molecules are aggregated in such. So that is why I asked it.
Absolutely. Thank you very much for this advice. But actually, we are exactly performing this kind of experiment with an experimental group here in Trieste, in order to, by mass spectroscopy, to find the composition of the soft corona.
Thank you very much. But your calculation is really great. And we also have one question from the chat.
Yes.
This is a question from Mikhail Fuldesu. Thank you so much for the contact presentation. I have a question for you. Can we combine multiple phosphates?
Multiple
Phosphates. Multiple phosphates.
Multiple
Okay, it's not possible. Okay, it's quite a more technical question. It's very technical because basically it's not possible if we speak about a common system, let's say. But you can combine different force fields if the derivation of the equation of the force field are the same. You cannot combine any kind of force field together. I don't know, probably my colleagues Domenico Marson will give you more detail about the difference between force field. I saw in the picture that Domenico make this, so I don't want to give you any spoiler about this presentation. But the question is a very good question, but basically it's not so let's say it's not so trivial combine different force field, but it's possible with some specification that probably Domenico will give you during the presentation.
Any other questions?